Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
So this is Richard Jacobs with the Finding Genius Podcast. I have a returning guest, Marilyn J. Roossinck. She's a professor of plant pathology and environmental microbiology at the Biology Center for Infectious Disease Dynamics, part of the Huck Institute of Life Sciences. She has her own lab, the Roossinck Lab. Her focus is on virus plant and virus fungus plant interactions and virus evolution and ecology. So, Marilyn, thanks for coming.
Sure, no problem.
Yeah, tell me a little bit about your background first. What got you into studying viruses and plants, and then we'll fast forward to present after that.
Okay. Well, I started in kind of a different trajectory in my life, but I took a microbiology course, and I was introduced to Lambda, the bacteriophage, and was the first virus I'd ever really knew anything about. So I was so impressed with how it was so clever. This little package of genes in a coat, protein coat. And so I decided to change my career and become a virologist. So I did my PhD on human virology, actually, on hepatitis B virus, but I didn't really want to stay in animal or human research because I felt like plants offered an area where you could do a lot more work. The hosts are really easy to come by. You can have, essentially, you can have 1,000 genetically identical hosts to do your work with, and they're inexpensive, and nobody minds when you put them in the blender either. So I just decided that I wanted to make a switch into plant virology, and so I studied that for a long time, mostly evolution work, and there's really nice systems in plants where you can do a lot of experimental evolution. And then I got involved in fungal viruses a little later on, and actually right now in my lab, which is kind of downsizing, I'm planning to retire. But my lab is only doing one project right now on a fungal virus, and we've kind of been winding up all the other stuff.
So is there any form of life or any organism that doesn't have a virus?
Not as far as I know. We have not explored all of life, so we can't say that unequivocally, but there's no known life forms that don't have.
I guess there's just some maybe that viruses haven't been found, but again, to everyone's knowledge, it looks like so far every form of life has a virus associated with it, right?
That's what I would predict.
Yeah, that's amazing. What do you think came first in evolution, viruses or cellular life, and why?
Well, I think viruses came first, but that's just speculation. We don't have any real evidence. But there is a whole hypothesis about the RNA world, which would imply viruses came first. Although there could have been RNA cells too. But yeah, it's my opinion that viruses are the original replicating forms on the planet, but others would disagree. So we don't, without a time machine, we really can't tell.
Do you think we'll ever know, or it's one of those questions that'll be forever unanswered?
Well, who knows? I think maybe we'll have a time machine someday.
Well, barring that.
It's not a question that's easy to answer because, it's just, there are people with pretty strong opinions, let's put it that way, but
yeah, I've seen that.
They don't have evidence to say they're right or wrong.
Okay. What role do you see that, this is a broad question, but what role do you see viruses have played in evolution and adaptation and even speciation? What roles, if any?
Oh, viruses are critical. Yeah. So, for one thing, 8% of our genome is retroviruses. That's just one kind of viruses. So they've been around with us since the beginning of human evolution, for sure. Many of those are shared with other primates, so they can be traced back for a long, long time. There are some older than primates. And in fact, there are some that go all the way back to things like They're shared with calocamp, which are very primitive life. Well, they're kind of fossil. They're considered living fossils. So, they've been with us for a long, long time. The roles that they've played in evolution, well, the most obvious roles are with retroviruses because they're in our genomes, and there are retroviral genes that are critical for our survival, like the evolution of placental mammals required a retrovirus. So they've been there and been involved certainly in things like speciation. A lot of this is just, we don't have fossil records of viruses themselves because they're too small and they're not maintained in, stone or things where we get fossil records from. But we do have this sort of paleovirology field where we look at the viruses that are integrated into genomes, and that's kind of a genetic fossil record. So yes, they've been around for a very long time, and I guess part of the thing that we have to think about is that, we're not really separate from our viruses. So our viruses are part of us. So to say that, it's just, you have to get a more holistic view, I think, of things. For example, a human being, what is a human being? Well, we are not just human cells. We have lots of bacteria. We have lots of viruses. We have lots of fungi. That's a holobiont. That's a single entity, encompasses all of those other things, including viruses.
Yeah, it's like we have a house with our family and our cousins and their kids and their families, and we've got a whole community living in us.
But it's a little bit. Symbiosis is a field of study, that's still actually needs a lot more work in it. But this understanding that some entities are more than one species, that really started in the mid 19th century, thinking about that, and it's still sometimes a hard concept for people to grasp. So I don't really like to think about viruses as if they were separate from their hosts, because they're really not.
Interesting. Okay. When I look at my dogs now, I think of them as, I think about their microbes and their viruses, and I see them in a little bit of a different way. It's weird. Like this is kind of a, an outside question, but since we're composed of all these things, we still feel like one being. It's weird. And how come we seem to have no sense of our constituencies and no sense of our makeup, that we feel like one creature? Like, any idea to you? Have you ever contemplated that, why we feel like we're one thing, but we're not?
Well, I don't think many people are actually conscious of all the different human cells they have. So I don't know why they would be conscious of the non-human cells. Biologically, yeah, we are one big symbiotic holobiont. That holobiont is the word that people use to say, all of the various constituents put together.
Right.
So
yeah.
You're not conscious of your lung cells or your heart cells or anything else. So why would you be conscious of your microbes?
That's true. Yeah. It would probably be overwhelming if I was, or if anyone was. Maybe that's why. I don't know.
Yeah.
But, very good.
You're mentally, you're not conscious.
Right, yeah.
Your body, your immune responses, who knows, but many parts of you are, the sort of autonomic parts of you are certainly aware.
Okay. Well, moving into some of the more specific virus questions, everyone tells me, virions are non-motile, they're just floating along and they're passive, and they don't really do anything until they come into contact with a cell. So, if I imagine them and they're like 50 to 100 nanometers, just ballpark average, and the hosts that they infect or are part of are huge, some are bacteria, maybe microns, some are people, a meter and a half tall. How do they, if you think about all time, I don't know how many infection events there have been. I'll just say, throw out a number like 10 to the 20th. I don't know. How could viruses so reliably find their targets and infect when they're so tiny and supposedly, incapable of sensing or doing anything? How do they find their way to the right spot so many times?
Well, every virus has a different mechanism for that. And that's one thing that it's difficult, I know, not to generalize, but you can't really generalize too much with viruses because they're all different and they all have found different ways to get into their hosts. So, it's not really It's not really quite like a big sea of viruses all around us, and once in a while they bump into the right host cell. They have active mechanisms. For example, most plant viruses are transmitted by insects. They can be transmitted by fungi and other things like farm machinery, but for a plant has a cell wall, and so for a virus to get into a plant cell, it has to make a hole in that cell wall. Insects do that very efficiently, so viruses are moved from one plant to another by insects. And the viruses actually have they control that process to some extent, because when they're infecting a plant, they will change the volatile composition of the plant, and the plant will start making volatiles that attract the insects to the plant, and then they pick up the virus and move it on to another plant. And they even go so far in some cases, as once an insect has landed on it and starts to feed, they take up the virus. Then the virus will induce the plant to make these anti-feeding compounds to encourage the insect to move off to a new plant. So the virus has manipulated that whole system to get itself moved around. And these relationships are very old and often very specific. So there are some viruses that are only transmitted by a few species of aphids, for example. And then there are others that are like generalists and are transmitted by hundreds of species of aphids. But that's just one example. But respiratory viruses, like we're dealing with now with this SARS-CoV-2 pandemic, they infect respiratory cells. In order to be an airborne virus, you have to be able to infect cells in the respiratory system. So then you just get breathed in, and there you go. You encounter your host cells. Those are two extremely different but important examples of how viruses move around.
Do you think that the viruses themselves are completely passive until they, through random motion, bump into the right cell, unless they're, like in the example you gave, specifically injected? But if they're not, if they're just near their targets, do you think there's any sensing going on and orientation and movement towards the right host cell for them, or is it passive at that point?
Directed movement like that, no, they don't have a way to do that. But on the other hand, if you're going to get infected with, we don't really know how many viruses it takes to get infected with this SARS-CoV-2, but if we look at something like influenza, you need about 10,000 or so virus particles in the tiny droplet that you breathe in. That's what it takes to establish an infection. So most of them fail. Very few of them actually are able to infect cells. And the reason that they're successful is just because they make so many hundreds of thousands of progeny virus. So they swamp the system, basically.
No, so you think the vast numbers of them is what gets them to the end goal, once they're at least in somewhat proximity to the host cells?
Right. So they do. In general, viruses need to have a lot of individuals in order to have establish an infection. And it might be that only one virus actually gets into a cell.
Well, okay, yeah. I feel like I want to ask you all the questions at once, but we'll step through them. We're getting there. Do you think viruses are alive? Why or why not? And it's just my speculation. I'm sitting there looking at a tree, and I'm thinking, okay, if there's a seed that made that tree, and if most of my experience was in seeing seeds and only every so often seeing a tree, I would think maybe trees are like viruses. They're mostly seeds and they're not alive. And when they're in the right conditions, now they're alive. But when I think about virions versus the form of the virus that's inside of a cell, perhaps it's the same thing. Maybe they're contingently alive once they enter a cell. Maybe they're just dormant, but they're alive the whole time as a virion, just like a seed is dormant and, becomes active in the right conditions. So with that context, like what's your thought?
There are people who think that. In fact, viruses were always assumed to be alive until they were first crystallized. So tobacco mosaic virus was the first virus that was crystallized, and then people said, oh, they make crystals. They're more like a chemical than a life form. That's where the whole debate started. So that was probably about 40 years or so into the field of pathology before this question really arose. Just to put it into historical context. But Why does it matter whether they're alive or not? Who cares?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
The reason why is that to me, if you assume that they're not alive, then you'd assume that they're incapable of certain behaviors or behavior at all. But if you do assume they're alive deliberately, then perhaps maybe your mind would say, "Oh, okay, well, these are some of the other questions I'm going to ask you." Most of them are predicated by. They could be predicated on, if you assume viruses are alive, you'd answer one way. If you assume they're not alive, you'd answer another way. So that's like the overarching reason why I asked. But that'll come out as we go through the questions.
I don't make any assumption. I just think it's not a really relevant. But you know that I already told you that before. So.
Okay, yeah, no problem. So do you believe that viruses have a group identity? If you look at viruses infecting something, it doesn't appear to be that there's just one specific genetic sequence of a given virus. It seems like there's, I guess, what's called quasi-species and different kinds with, different variants. Do you think that a virus is able to act in a way where it knows self versus other? And do you think that, all the quasi-species of a virus, when it infects, kind of act in concert with different abilities? Just like a bee colony, has phenotypically different bees, but they're all still bees and they all act together?
I don't think viruses have a sense of self, because that's something that requires a higher order of consciousness. And I don't really think viruses are conscious. But a quasi-species, you brought up a quasi-species, so let me explain a little bit about what it is. It's a term that comes from physics and not biology, so it's an unfortunate term. It has nothing to do with the biological concept of species. So a quasi-species is a word that's used frequently for RNA viruses in particular. Some DNA viruses, however, behave this way too. And all it is that when a virus infects a host and begins to replicate, it makes a lot of mistakes in its replication process, and they're not usually corrected. Some viruses can correct them, some can't, but this generates a very diverse population. And that diverse population within a single host, or perhaps within a single host cell, that is a quasi-species. And it does not act, they do not act as individuals, they act as an entity. The quasi-species is the entity. So they complement each other. Some may be defective, some may have different functions in different parts of the genome because of this rapid evolutionary process where they're making so many mistakes in their copying of the genome. And so that they do act as a single entity. So it's like it just has a lot of variation of its genes. You can, if you know anything about genetics, you know that we generally have 2 alleles, sometimes we have more for each gene. Well, you can think of a quasi-species as just an entity that has thousands of alleles. That's essentially what a quasi-species is. So it has to be infecting, it's a single replicating population. That's another part of the definition. So you're not going to have, it's in one host, it's in one cell. You don't have a quasi-species spread out among a bunch of hosts. It's a single population.
Well, if there's a virus and, I don't know, let's say I'm infected with it and I label myself number 1, and then I infect someone else and they're number 2 and someone else number 3, by the time number 20 gets its passage through 20 different hosts, again, me or whatever species you want, what do you think will be different about the virus that goes after number 20? Do you think it would be more commensal, more virulent, less virulent? What changes do you think would occur and why?
Well, 20 isn't very many, but so probably not much of a change in that small amount of transmission. But in general, let's say for longer term infections, you would expect a virus to evolve to be less pathogenic. And in fact, there's a whole field of, the evolution of virulence that's been pretty extensively studied. The hypothesis is that a virus evolves to become less virulent because virulence is generally not an advantage. So a virus only wants to do one thing, and that's replicate. That's really the goal of a virus, if it had a goal. It's just to make copies of itself. And if it's making its host so sick that the host dies or even stays home and can't get out and visit its friends, that's not an advantage for the virus. And if it kills its host, pretty soon it won't have any hosts left to infect. So that would be a really bad thing for it. So in general, you think that viruses probably evolve over time to be less pathogenic. The other thing you have to remember we talked about how it might take 10,000 viruses to infect one cell. So that what that means is that, okay, you have this population of 10,000 viruses that came from a simple original quasi-species, but only if one or two of those or a few of those are going to infect the next host. So there you're going through a population bottleneck. And so there's going to be drift in that virus from host to host because of that bottleneck. Only a few members of the original quasi-species are going to get into the next host. And that number can vary a lot. So I don't really know that much about the multiplicity of infection for various viruses. I know that it's quite different in different viruses. So some viruses may have a multiplicity of infection of, thousands, and some may have only a few. But that's going to impact that whole process of evolution and adaptation to the host as well.
Yeah, in some of the examples you gave, the viruses have like very, very specific ways of being, transmitted between hosts. And I don't know if there's an analog in plants, but when I look at some of the viruses that affect people, like rabies or influenza or HPV, it seems like these cells that the virus targets and infects are also the same cells that enable it to spread from there. Influenza, my respiratory cells are affected, so I cough and sneeze, and it spreads that way. Why does there appear to be, at least in some cases, this matching of tropism versus method of spread? Why wouldn't I get, why wouldn't H? Good.
It's always matching. I mean, if it doesn't match, you wouldn't be able to establish an infection.
Well, what if I have a sexually transmitted disease? Why can't I spread it by coughing or sneezing? Why wouldn't it?
It doesn't infect the respiratory cells.
Right. It's just strange that it seems like there's this matching. I just wonder why it's maybe it's just a just so story. I don't know.
Well, I think what you have to remember is that all these are happenstance relationships. And so it's just, it's an accidental initially, it's a happenstance. The SARS-CoV-2 happens to have this protein that attaches to the ACE2 receptor in human respiratory cells, right? So that was a happenstance, or perhaps it derived from its previous host, which we don't really know what that was yet. But because of that, now the virus can infect respiratory cells. But if a virus doesn't have a way to infect a respiratory cell, it doesn't matter how many particles of it you have that are floating around in the air, you're never going to get infected that way. So it depends on it all goes together. I mean, these evolutionary events didn't happen overnight. These viruses have been around for a very long time. And even SARS-CoV-2, yes, it's only been in humans for a short time, but it's been in whatever its animal host was for probably thousands of years. So yeah, they have evolved different ways. You mentioned rabies, because rabies, at least in some hosts, it does change behavior. So it is a virus that infects the nervous system, and it can change behavior to biting, but that's not the only way it changes behavior. In fact, in cattle, when a cow has rabies, they start choking, and so the farmer puts his hand down the cow's throat to see if he can figure out why the cow's choking, gets a little nick in his hand, and gets rabies virus that way. So that's a different kind of behavior that rabies can induce, which also aids in its transmission. And there are many examples of viruses that affect the behavior. We already talked about the ones that change the volatiles in plants.
Yeah, I was going to ask you, to what level of control does a virus exert over a host cell? I know it uses cellular machinery to replicate itself, but how could it cause a plant to change the volatiles it expresses to attract insects to spread it, or to keep insects away once it reaches a certain point? How could that be?
Well, it's just a matter of genetic control. It has we don't really know the details of how that works yet. Those are fairly recent studies. But the virus has some way of turning on a gene or turning off a gene in a plant. And That's often done by small RNAs. They're major controllers of genes, really important in genetic regulation. But we don't know the details of that with that particular system. But nevertheless, they are, if it's going to be an advantage, then it'll probably happen. If the virus had no way of helping the insects find the infected plant, then its transmission would be pretty poor. But once a virus figures out how to do that, well, it's not that the virus figures out. Once the virus makes something and that's where the happenstance comes in. So somehow the virus made something that turned on the volatile emissions in this plant. And once that's happened, then all the progeny viruses after that are going to have that feature because it's a huge advantage. So the initial thing that happens is an accident. But if it's an accident that's an advantage, then it becomes predominant. That makes sense?
Yeah. So do you think that viruses are able to have cells send out, let's say, specific extracellular vesicles to affect other cells, or to get bacteria to send out certain specific plasmids? Do you think that they're able to, let's say, do a quorum sensing and coordinate action by using cellular machinery or bacterial machinery to signal other infected bacteria or cells?
Yeah, I'm not really very up to date on bacterial physiology, but I do know that viruses, for sure, bacterial viruses affect the bacterial host in many different ways. Most of the time, they're an advantage for bacteria. So if they're a lysogenic virus, that means they've integrated into the genome of the bacteria. Then that provides an immunity so that that bacteria cannot get infected with the lytic form of that virus. It can also, as far as a population of bacteria goes, it can become lytic, in other words, release virus particles that can wipe out competitor bacteria. So that's a way that it affects the outcome of a bacterial population, say. But yes, they also encode virulence factors. I think cholera is one example. The cholera phage, there's actually two of them that produce cholera toxin. And without those phage, cholera cannot invade the human gut. We have a lot of cholera in this country. The Chesapeake Bay is full of cholera, but we don't have any of the phage. So it's not a cholera that can cause the disease. And there are many other human pathogens. Diphtheria is essentially caused by a bacteriophage in the diphtheria bacterium. So yeah, there are lots and lots of examples of that. So it is.
Yeah, beyond again co-opting cellular machinery to produce more of the virus, whatever virus it is. Again, do you think that the control over the cellular machinery extends to, again, things such as cell-to-cell communication, whether in bacteria or in plant cells or in human cells? Can, again, the virus instruct the cell to send out extracellular vesicles that have certain packaging in them? Do you think it can use the cell to, again, communicate with other cells and do quorum sensing? How many cells of this type are infected by me, at one time?
I can't tell you about quorum sensing. I don't know very much about it. But certainly viruses can induce signals to other cells. In plants, we know this happens in plants and in fact, there are some recent examples of plant viruses that have multiple components. They have many different segments of their genome, and you don't have to actually have every segment of the virus genome in every cell of the plant. So in other words, this plant cells in a little focus of infection can cooperate and share viral gene products among those cells. So yeah, definitely things move between. Interesting.
Why do you think that there's a latency period from the time an organism is infected till it shows, pathogenicity or problems? Do you think it's just a matter of time that the virus needs to replicate enough and affect enough cells, or is there other reasons?
Well, it's probably mostly to do with the virus establishing an infection. So if the virus infects like one or two of your cells, it's going to take a little time for it to ramp up and infect enough cells so that there are symptoms apparent. So yeah, it's just a matter of ramping up its replication.
What do you think governs, latent versus, lytic behavior, or lysogenic versus lytic behavior, versus endogenization? Why would some viruses, let's say, infect and never really seem to cause any problems to a host for months, years, days, lifetime, and the host encounters stress, and now maybe the virus turns lytic? Why do you think that might happen?
Well, there could be a lot of different reasons. I mean, then again, don't generalize so much, because it's probably different for every example you can come up with. So there are viruses. I mean, these terms are not very well defined often. Latency versus persistence versus acute. All these things I've tried to define them in my own systems, because they're often used in different ways in different host systems. So there are viruses that are called latent, which means that there's no obvious effect on the host. It doesn't mean there isn't an effect. It's just that we haven't found an effect. And they can convert, but to becoming acute, in animal systems in particular, plants have persistent viruses that never convert to acute viruses. So that's a pretty different system. And in fungi, there are also most viruses are what I would call persistent in fungi, and they may have an effect under the right environmental conditions. So sometimes they have very dramatic effects. They can be very beneficial. For example, we studied a virus from Yellowstone National Park that conferred heat tolerance to the plants. So the virus infected a fungus. The fungus colonized the plants. And if you had all three together, they could grow in soil temperatures of over 50 degrees centigrade, which is pretty hot for a plant. But the virus had to be there. The fungus couldn't do it by itself. So we don't know how that relationship was established. We have looked at a lot of, it's probably pretty old. Yellowstone has been around for about 600,000 years or so. So
wow.
When those relationships got established in that timeline, we don't really know. But again, it's almost certainly happenstance. The virus happened to make something that helped confer this thermal tolerance. And then once that happened, then the only plants that could survive were the ones that had the fungus and the virus. So a lot of selection there to keep the virus around.
If I had a plant cell or an animal cell or any bacteria, and I sucked out a bunch of the contents where it was not really functional, but the membrane or the cell wall was still intact, do you think viruses that do come and fuse and enter, for instance, those kinds, do you think they still would do that, and they would just, or would they stop and sense something's wrong and midway stop infecting?
Well, I don't know. It depends on what's left. The virus does require certain elements of the cell, like it has to have membranes. Most viruses use membranes for replication machinery. It has to have the protein synthesizing machinery. I mean, the virus infection can't get anywhere without those things. And that's the one thing that all viruses seem to need is the ribosomes, the protein synthesis, and generation of energy by the cell. So if you didn't, if you don't leave anything in the cell, then no, the virus can't infect it. But the virus isn't going to say, oh, this is a bad cell, I'm not going to infect it. I don't think so. I mean, the way a virus enters a cell is very tightly programmed, depending on the virus and the host. But it's usually involves cell receptors. In most human viruses require a specific receptor on the cell surface. If that's there, the virus will probably get into the cell. But I don't think a virus knows the difference, basically. If the receptor's there, then it's there.
Yeah, so maybe in the future, if cells could be made with a faithful membrane or cell wall, they could be traps for viruses. The viruses that do fuse and enter would do so when no one's home. Now they're stuck.
Yeah, I guess that's a possible strategy. I hadn't really ever thought about it.
Is there a one virus, one host cell model, or are there some where you need to have multiple viruses that coordinate action to infect the cell or are necessary to infect the cell?
Oh yeah, there are lots of examples of that. I know of examples in both plants and fungi where you have to have more than one virus. Sometimes they're called helper viruses. So one virus provides part of the function and the other virus provides another part. And then also the other thing is that there are a lot of plant viruses and fungal viruses. That have divided genomes. They tend to package each genome segment in a separate virus particle. So those are kind of like individual, components of the same virus, but you have to have, if you have 4 or 5 different components, they all have to get into the same cell in order to establish an infection.
Which virus does that? What's the specifics of that example?
Well, let's see. Cucumber mosaic virus has 3 particles. Some of the nanoviruses have 8 to 10 particles. Those are small DNA viruses that I don't know a lot about, but so from 2 to 10 is what about the range of number of particles, and there may be some others that are a little more, but many viruses of plants are like that. And presumably that's a strategy that evolved because plant viruses in general are very small, and except for algal viruses, all other plant viruses are very small. And the reason we think that they're so small is because they have to move between the plant cells through these very restricted openings. So that may be why they require like 3 virus particles to make the full complement of the genome. That allows them to stay really small.
Yeah, that's amazing. So in the case of a plant virus that does have multiple virus particles that each contain part of the whole payload, is there an order in which they infect? Are they all affect at the same time? Like, does one arrive and then it waits for others? Like, what's known?
As far as we know, they probably all have to get there around the same time. They're not going to, the infection isn't going to go very far without all of them. So probably you get multiples of each. I mean, it's very unlikely that you would just get exactly 3 of the, one of each of the 3 different particles, for example, into a cell. You'd have multiples of some of them just by chance. So
okay.
There are also some other very interesting systems. Like there's a recently described virus in fungi where there's 2 very different viruses that are dependent on each other for infection. So sometimes in those cases you could have one virus, like you could have virus A without virus B, but you can't have virus B without virus A. And some of those, if we look at the genomes of viruses, we can see that those kind of interactions probably happened in their evolutionary history quite a bit, and then some parts were lost to create a new virus that just had the essential parts. There's actually a really good example of that in plants, where it used to be called P. nation mosaic virus, and now I think there are 2 different viruses, but essentially that's an example where you have 2 separate viruses, but they have to co-infect, and one of them provides the movement protein for the other one, and so it can move into different parts of the host. It's a ludio in the family called the ludioviruses, which normally only infect the phloem of plants, the vasculature of the plants, but because this virus has the other virus with it, this can infect all of the plant tissue, all the mesophyll cells anyway. So and I would assume that if we could take a time machine forward now, we probably wouldn't be able to find that virus in the future. We would just find one where all the extra redundant parts had been lost.
So some viruses will infect, and then I guess they prevent superinfection, and then you have examples of ones that need other viruses in order to complete the infection.
Mm-hmm. Well, superinfection usually refers to the same virus. So, for example, there are lots of examples of this, although I have never studied any of them myself. But for example, if you have a cell that's infected with virus A, it's not very likely that it will prevent infection by virus B, but it will prevent infection by other virus A strains. But you see mixed infections in all kinds of things. There's a phenomenon that's called viral interference. It's not really very well studied. In some cases, it's pretty well studied, but so sometimes if you have a viral infection, it will inhibit another virus infection. And part of that might also just be, for example, if you have influenza virus, your innate immune response is going to be kicked into high gear, and that might prevent infection by any other virus at that point. So that's a different thing than at the cellular level. At the cellular level, mixed infections are not uncommon, but usually if you have a particular virus in the cell, it may exclude other members of that same virus.
That's true. Yeah, there's so many coincident factors, it may be impossible to pick that apart inside of a living organism.
Mm-hmm. Yeah, that's the problem. And they really frown on you doing experiments on humans, and please don't quote me on that.
I gotcha.
I'm just joking. It's not, the sad truth is it's been done on quite a lot. Human experiments have been done quite a bit.
Yeah, well, that's true. Do you think, if you consider, I don't know, plants or bacteria or people, does their phagome or virome, do you think, really impact their immunity? Is it part of their immunity, or what do you think the relationship is there?
Oh, certainly, that's part of it. And there have been some nice studies done, not in, we sort of extrapolate things to humans when we find them in other animals. But for example, mucous membranes are often covered with bacteriophage from the microbiome of the organism. And of course, mucous membranes are places where we have entry into the organism. And so it's thought that those are there to provide the first line of defense, so that if an incoming bacteria is coming through past this mucous membrane, the phage that are there will attack it. So that's kind of like another level of immunity that viruses can provide. Yeah, so, and they are all, we just don't know all of the interactions. The whole idea of the virome is fairly recent, and I think we may have talked about this before, I can't remember, but there's a lot of people who think viruses are all bad and evil, and that's sadly misinformed. They're sadly misinformed. So most viruses are actually beneficial or commensal. The pathogens are the rare viruses, but they're the ones that everybody studies.
Yeah, no, that's true. I guess one really way out question, but if you were to, how they use like AI for drug discovery, and they're trying to find drugs that will fit this receptor or how this protein folds, etc. Right. Do you think there would be a way to use viruses because of the how fast some of them can mutate to explore some kind of biological information space? Put them in with a given host cell and put the host cell under different stresses and see if the viruses mutate and now are able to enter when they couldn't enter before, or vice versa. Do you think they could be used in such a way?
Oh, sure. I think that's actually quite possible. And it's actually something I wanted to do, some experiments of just to try to get a virus to evolve to infect a different kind of host, for example. I just never got around to it. We all have lots of ideas that we never can actually bring to fruition. But I think it's very possible that, the evolutionary capacity of viruses is very rarely appreciated. So I'll tell you an experiment I did in my lab. We were using a satellite RNA. It's a small parasitic RNA that sometimes they are replicated by plant viruses. And in this system, I could get in 10 days, I could get 2% of the nucleotides to change just by changing the helper virus. So I changed the environment of that small parasitic RNA, which is replicated by a viral polymerase, and 2% of the nucleotides changed in 10 days. And that's pretty phenomenal, because that's like the difference between our genes and chimpanzees' genes, That's a pretty big leap. And that happened in 10 days. So I just tell you that to illustrate how rapidly viruses can evolve under the right selection pressure, because they have that huge mutational capacity. So yes, I think this could be exploited more.
Okay. Well, very good. Any last thoughts that you want to share? Things in virology that is there anything that's like astounded you that you've never had time to really investigate more fully, or simply that you just couldn't believe that you saw this in viral behavior?
There are many things I wish I had time to investigate, that's for sure. I don't think any of us ever get to do all the things we have ideas for. But I guess my last message would be, I really hope that people begin to appreciate the value of viruses, how important they've been to the evolution of life on Earth, and how many of them are beneficial, and how we need to understand that beneficial nature of viruses better. And that's where viruses could likely be exploited, is just understanding how that kind of thing works. Yeah, so that's my last word. Let's go for the good viruses.
We need a T-shirt that says, "Have you hugged your commensal viruses today?" That might be good. So,
yeah.
Well, very good, Marilyn. What's the best way? You said you're winding down. You're getting closer to retirement. But for interested folks, how can they find out more about you? Where can they go?
Well, I do still have a website, roossinklab.com. Not sure how much longer I'm going to maintain that, but to be honest with you, a Google search with my name will get you lots of information. And we are still writing up and publishing papers, so
excellent.
Ongoing for a while.
Well, very good. Well, thank you for coming on the podcast. I appreciate it.
Hey, thank you. Good luck with the book.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.